All right, let's jump right into one of the biggest, most fascinating puzzles in all of biology. You know, every single cell in your body, from a neuron in your brain to a cell in your skin, has the exact same DNA, the same genetic blueprint. So how? How does one become a nerve cell and the other a muscle cell? Let's unlock how that gene control actually works.
I mean, this is really the core question, isn't it? If the instruction manual is identical for every cell, how do you get these completely different results? A nerve cell is built for sending signals, a muscle cell for contracting. Their jobs, their shapes, they're worlds apart, but they're built from the very same set of instructions. So the mystery isn't what's in the book, it's about who's reading it and which chapters they decide to focus on.
And there it is. That's the secret sauce. The difference isn't the genes themselves, but this whole other layer on top called gene regulation. It's this incredibly sophisticated system that's constantly deciding which genes get turned on, which get turned off, and even how loud their volume is. This is what gives every single cell its unique identity.
Okay, so to get a handle on this control system, we need to meet the main characters. I want you to think of them as the directors of a giant, sprawling genetic orchestra. These are the proteins calling all the shots, deciding which part of the musical score, the DNA, actually gets played. So these directors, they have a formal name, regulatory transcription factors. We'll just call them RTFs. Their whole job, their entire purpose, is to grab onto specific parts of the DNA and either crank up the gene reading process, which is called transcription, or dial it way down, or even shut it off completely. They're the conductors, making sure each section plays its part just right.
Now, it's super important to make a distinction here. Our RTF directors aren't the only players. There's another group called general transcription factors, or GTFs. Think of it like this. The GTFs are the stage crew. You absolutely need them. They set up the stage, they get the lights on, they get the basic machinery ready for any gene to be read. But the RTFs, they're the ones with the baton, telling the string section to swell up now while the brass section stays quiet. They provide all the nuance and the specific control. So, how in the world do these director proteins actually read the DNA blueprint? I mean, how do they find the right page in a genetic library that has 3 billion letters in it? Well, it all comes down to some really elegant molecular chemistry and basically a perfect physical fit.
Yeah, this is where the real magic happens at the molecular level. DNA is just this incredibly long sequence of 4 chemical bases. How does a protein sift through millions and millions of them and lock onto the one specific sequence it's looking for? The answer is all about shape. It's geometry. You see, many of these RTF proteins have a special structure that looks like a spiral staircase called an alpha helix. And it just so happens that this helix is the perfect size and shape to slot right into a specific groove and the DNA double helix. It's literally like a key fitting into a lock. This snug fit allows the protein to get close enough to form chemical bonds with the DNA bases and essentially read that specific sequence. And these directors have a whole toolkit of these structural shapes or motifs to do their jobs. There's the helix-turn-helix, which is a super common one. Others are called zinc fingers, and they literally use a zinc atom to hold their shape together so they can bind to DNA. And then there are really cool ones like the leucine zipper, whose job is to help 2 of these director proteins team up and work together as a pair, which is often crucial for them to function. Okay, so we've got our directors, the RTFs. But where on the DNA do they actually land? This is where we introduce the remote control panels they use to operate all this genetic machinery. These control panels are actually regions of DNA called enhancers. An enhancer is pretty much just a stretch of DNA that has these specific docking sites or landing pads that our RTF proteins are built to recognize. It's the exact spot on the blueprint where the director knows to make their mark.
And this is where things get really wild. What's so incredible about these enhancers is how flexible they are. Get this, they don't have to be anywhere near the gene they control. They can be thousands, even 100,000 DNA letters away. To put that into perspective, that's like having a light switch in your house that controls a lamp in the next town over. They can be before the gene, after the gene, even tucked away inside another gene. Oh, and they can be flipped completely backward and they still work. This is true remote control. All right, so the director, our RTF, binds to its control panel, the enhancer. What happens next? Well, now it's time to either hit the gas or slam on the brakes. And this is the work of 2 different flavors of RTFs, activators and repressors.
Let's walk through what happens when a gene gets turned on. First, an activator protein finds and binds to its enhancer, which, remember, could be super far away. Then, these other proteins come in and cause the DNA to literally bend into a loop, which brings that distant activator right up close to the gene's starting line. Now that it's nearby, the activator can give the whole transcription machine a huge kickstart, boosting the rate of gene expression by up to 1,000 times. That's called upregulation.
And you can probably guess the opposite happens too. A repressor protein can bind to a spot on the DNA, sometimes called a silencer, and do the exact reverse. It can physically block the machinery, get in the way, and just slam the brakes on transcription. And that, of course, is downregulation.
This brings us to the final, and maybe most mind-bending layer of this whole puzzle. We've got these incredibly powerful director proteins controlling our genes. But what controls them? Who directs the directors? Because they can't just be running around active all the time in every cell.
Well, the cell has a few really clever ways to keep its RTFs in check. First, a small molecule, like a hormone, can act like a key. It binds to the RTF, changes its shape, and suddenly it can bind to DNA. Second, as we saw earlier, sometimes these proteins have to team up with a partner to get the job done. And third, the cell can just stick a little chemical tag, like a phosphate group, onto the RTF, which acts like a simple on-off switch.
So, what this all comes down to, when you put it all together, is this beautiful concept called combinatorial control. It's not one single switch that makes a cell a neuron. It's the unique combination, the specific cocktail of active RTFs, available enhancers, and signaling molecules that all work together to write a totally unique script for that one cell type.
And this quote from Robert Brooker really just nails it, bringing us right back to where we started. It perfectly sums up everything we've talked about. The incredible diversity of cells in our bodies isn't because they have different DNA, but because of the masterful, elegant, and super complex regulation of that DNA. And that really leaves us with a pretty mind-blowing thought for the future. We're just beginning to decipher this incredibly complex regulatory language. But if we can truly understand all the directors, all the control panels, all the switches, could we one day learn to write or maybe even rewrite our own cellular scripts? Imagine being able to tell cells how to regenerate tissues or fight off disease. The possibilities are as complex and as profound as life itself.